These rivers and streams taught me to dream. The mountains and trees are comforting me. I've tasted the world, but nothing is sweet as all our memories. You're my haven, always there to pick me up when I need to be whole again. There's nothing quite like you, rivers and skies so blue. So I keep coming back to you. Shy and so beautiful, you never leave me cold. So I keep coming back to you. The songs in the caves bring back the heartache. I know how I miss the wine from the hills. Bonfires lit, pour me a drink, love. Let's dream away. You're my haven, always there to pick me up when I need to be whole again. There's nothing quite like you, rivers and skies so blue. So I keep coming back to you. Shy and so beautiful, you never leave me cold. So I keep coming back to you. Back to you.
Spoštovani gospe in gospodje, cenjeni predavatelji in govorci, spoštovani udeleženci, kot tudi vsi, ki nas spremljate v neposrednem prenosu prek spleta, v imenu organizatorjev in sponzorjev vas prijazno pozdravljam na današnjem javnem forumu o Alzheimerjevi bolezni. Gre za zaključni dogodek 15. mednarodne konference ICBEM 2024 v Ljubljani, ki je tukaj v Grand Hotelu Union zbral najvidnejše svetovne strokovnjake, znanstvenike in raziskovalce na področju energijske presnove v možganih, s posebnim poudarkom na preprečevanju ključnih bolezenskih in starostnih sprememb v možganih v prihodnje. Uvodoma smo videli promocijski video Slovenije, avtor studio Pirc, glasba Maja Kevc, vir www.slovenija.info. Nadaljevali pa bomo v angleščini, ki je uradni jezik konference. Honored guests, ladies and gentlemen, it is with pleasure and delight that I welcome you all to today's public forum on Alzheimer's disease in the Grand Hotel Union in Ljubljana, the final event of the 15th International Conference on Brain Energy Metabolism, ICBEM 2024, which has brought together leading researchers, clinicians, young scientists, and students all interested in brain energy metabolism. We have been given a unique opportunity to offer you hospitality in Slovenia and have been truly privileged to benefit from your vast knowledge and professional expertise. Our warm greeting on behalf of the organizers and sponsors goes to our distinguished guests, Your Excellencies ambassadors, representatives of the Spominčica Alzheimer Slovenia Association, heads of clinics, University Medical Center Ljubljana, representatives of the government of the Republic of Slovenia, Keynote speaker, Professor Sir John Hardy, UC Institute of Neurology, London, UK, and Dr. Aleš Musar, spouse of the President of the Republic of Slovenia, Dr. Nataša Pirc Musar. September is World Alzheimer's Month, and today's public forum coincides with World Alzheimer's Day, which is celebrated each year on September the 21st. On this day, the world concentrates its efforts on creating awareness of Alzheimer's disease. The disease, as we know, is among the most prevalent forms of dementia, a set of disorders that disrupt mental function. World Alzheimer's Day is therefore the perfect opportunity for us to raise our voices and find new ways of countering the disease's effects. The main purpose of today's public forum is to sum up the latest scientific developments in the understanding of mechanisms responsible for neurodegeneration and impairments in brain energy during aging, and present them to the wider interested public. Considering the fact that human brain uses a lot of energy when compared to the energy consumption in the rest of the body, as we can see on the slide, the prime importance of relevant neuroscientific studies for a better quality of our lives easily becomes transparent to us all. Let us spark off the highly significant theme of today's public forum with a quote by co-discoverer of DNA structure and pioneer of molecular biology, Dr. Francis Crick, that I quote, "There is no scientific study more vital to man than the study of his brain. Our entire view of the universe depends on it." End of quote. Ladies and gentlemen, dear viewers, for the first address tonight, let us warmly welcome ICBEM 2024 co-chair academician, Professor Dr. Robert Zorec. Prijazno vabljeni.
Many thanks. The program committee members are from all corners of the world and worked very hard to prepare an inclusive scientific agenda. Many thanks to each and every one of them. Having ICBEM 2024 in Slovenia is an acknowledgement and support for science in Slovenia, and also a stimulus to address the challenge of containing neurodegenerative diseases in the country, a local and global challenge. Although 2 new drugs have been approved recently by FDA, but not by EMA yet, to treat Alzheimer's disease after 25 years, these are not ideal, exhibiting serious adverse events. However, this generated new commitments to develop new ones, and ICBEM 2024 participants discussed possible targets for new therapy developments. In Slovenia, the rate of aging is among the highest in the EU. With this, the burden of age-related cognitive decline is significant. About 5% of the population, patients and caregivers, is engaged with this problem. With ICBEM 2024 in Slovenia, we are encouraged to contribute to solving the global challenge of neurodegenerative diseases. Thank you.
This has been short and sweet. Thank you. We kindly thank academician Professor Dr. Robert Zorec for focusing on the main findings of the ICBEM 2024 conference in Slovenia and for evaluating the present age-related cognitive decline situation in our country. And now, ladies and gentlemen, dear guests, for a very special honor, our public forum on Alzheimer's disease and its participants have been granted by our next distinguished speaker, the President of the Republic of Slovenia, Dr. Nataša Pirc Musar, who is now to address us directly from the presidential palace.
Spoštovane gospe in gospodje, ob Mednarodnem dnevu Alzheimerjeve bolezni naj se vam zahvalim in podprem vaša prizadevanja na področju raziskovanja te zahrbtne in neozdravljive bolezni ter izrazim hvaležnost, ker pomagate osebam z demenco. Zahvaljujem se vsem znanstvenikom in strokovnjakom, ki raziskujete vse oblike demence in še posebej Alzheimerjevo demenco, ki je najbolj pogosta med demencami in obenem iščete ne samo zdravila za lajšanje simptomov, ampak tudi raziskujete, kako je mogoče to bolezen preprečiti oziroma jo pomakniti v čim višjo starost. Zavzemam se za dostojno starost, ki mora biti aktivna in zdrava. Demografska gibanja, ki so posledica napredka medicine in podaljševanja pričakovane starosti, nam kažejo, da bo delež starejših v populaciji vedno večji. To je neizpodbitno dejstvo. Veliko je odvisno od vsakega posameznika in njegovega zdravega življenjskega sloga, a žal svoje doda tudi genetika in drugi faktorji, zato je raziskovanje izjemno pomembno. Odkrivate nove diagnostične metode za zgodnje prepoznavanje demence in nova zdravila, ki so bolj učinkovita pri blaženju simptomov in upočasnitvi napredovanja bolezni, če so predpisana v zgodnjih fazah te nevrodegenerativne bolezni. Alzheimerjeva bolezen je že dolgo priznana kot bolezen in ne velja več, da je le del normalnega staranja, zato je tako zelo pomembno, da vlagamo v preventivo, preprečevanje, diagnostiko in zdravljenje simptomov. Glasgowska deklaracija, sprejeta na konferenci Alzheimer Europe leta 2014, je jasno začrtala človekove pravice oseb z demenco in nas zavezala k temu, da naj demenca postane prioriteta javnega zdravstva v celotni Evropi. V Sloveniji se lahko pohvalimo z odličnimi strokovnjaki in aktivno civilno družbo, ki so ji prisluhnili tudi odločevalci in ponosni smo lahko na vsak napredek na področju ozaveščanja o tej bolezni. Dokaz za to je tudi vaše današnje srečanje, saj se glas o uspehih naših strokovnjakov širi tudi v tujino. Tudi predsedniška palača je demenci prijazna in dobro sodelujemo z našim cenjenim društvom Spominčica Alzheimer Slovenija. Z njihovo pomočjo smo usposobili zaposlene za primerno komunikacijo z osebami z demenco, ki bi se morda zatekle v našo palačo. Strategijo obvladovanja demence v Sloveniji do leta 2030 smo postavili temelje sistema, v katerem bo vsaka oseba z demenco obdana z razumevanjem in bo imela dostop do kakovostne, dostopne in učinkovite obravnave. Načrtovani ukrepi bodo imeli občuten vpliv na izboljšanje kakovosti življenja oseb z demenco, njihovih svojcev in oskrbovalcev. Zavedamo se nujnosti, da se moramo dobro pripraviti na zdravstvene, družbene in ne nazadnje tudi ekonomske izzive povezane s staranjem in demencami. Demenca je povsod okoli nas in vsi se moramo naučiti živeti z njo. Smo na dobri poti tudi z vašo pomočjo. Zato še enkrat iskrena hvala za vaše neprecenljivo delo in še veliko uspehov v dobro vseh nas vam želim tudi v prihodnje. Srečno.
Our sincere thanks to the President of the Republic of Slovenia, Dr. Nataša Pirc Musar, for having expressed her support of the efforts and achievements of neuroscience today in view of our population's pressing problems and dilemmas, and for having shared with us her encouraging thoughts and messages. And now it is my pleasure to invite on stage ICBEM 2024 co-chair, Professor Dr. Kelly Drew, University of Alaska, Fairbanks, who has kindly accepted the task of introducing to us our esteemed guest, Professor Sir John Hardy, UCL Institute of Neurology, UK, and his lecture we are truly looking forward to.
Thank you. It is an honor to introduce Professor Sir John Hardy, a pioneer geneticist who has made significant contributions to our understanding of Alzheimer's disease. Sir Hardy's research has guided diagnosis and the development of new therapies to decrease the burden of this devastating disease. Sir Hardy was the first to identify a mutation in what is called the amyloid precursor protein gene. He found this mutation in families with early onset Alzheimer's disease. This discovery was crucial as it established a direct link between genetic mutations and Alzheimer's. Sir Hardy found that the amyloid beta protein can clump together and form sticky plaques in the brain, which are common in people with Alzheimer's disease. He then came up with the idea called the amyloid cascade hypothesis. This idea says that the buildup of amyloid plaques in the brain starts a chain reaction that eventually leads to dementia. This hypothesis has become foundational in Alzheimer's research and has guided therapeutic strategies aimed at targeting the buildup of amyloid in the brain. So Hardy's research also extended to what are known as tau proteins. He linked mutations in the tau gene to neurofibrillary tangles seen in frontotemporal dementia. This work further illustrated the complex interplay between different proteins involved in neurodegeneration and expanded the understanding of Alzheimer's pathology beyond amyloid. Sir Hardy's findings have paved the way for multiple drug developments targeting amyloid beta, including recent advances like lecanemab, which has shown promise in slowing cognitive decline in Alzheimer's patients. This represents a significant milestone as it translates decades of research into clinical applications. In recognition of Dr. Hardy's milestone, Dr. Hardy's contribution to the field of Alzheimer's research and molecular genetics, he was knighted by Queen Elizabeth II for services to human health in improving understanding of dementia and neurodegenerative diseases. His knighthood adds to a long list of honors that reflect his influence and leadership in neuroscience, marking him as a key figure in advancing our understanding of dementia. Professor Hardy holds position as Chair of Molecular Biology and Neurological Diseases at University College London and is a member of the UK Dementia Research Institute. He has received numerous accolades throughout his career, including being elected a Fellow of the Royal Society in 2009 and winning prestigious awards such as the Breakthrough Prize in Life Sciences and the Brain Prize. Dr. Hardy continues to look at how our genes and things around us, like what we eat or how much we exercise, can affect our chances of getting diseases like Alzheimer's. While scientists continue to look at other things that might cause Alzheimer's, like different proteins and how the brain gets inflamed, Sir Hardy's pioneering work and the development of new treatments guided by this work is a first step toward finding a cure for Alzheimer's disease and related dementias. Please join me in welcoming Dr. Hardy, who will speak to us today about Alzheimer's disease, a challenge for the future.
The quest for medical treatments and cures isn't just about slides and microscopes and what can you do about the symptoms. It's about families, it's about individuals, it's about stories of people who suddenly begin to lose themselves in a way that's frightening for them and terribly painful for those who love them.
A lot of people would say that Alzheimer's disease research, to some extent, started with Carol's family.
all of them were between the ages of 54 and 58. And it was at that stage that our life changed, really.
We put an advert in 1985, 1986 in the Alzheimer's Society newsletter because we were interested in studying the genetics of Alzheimer's disease. And the very first letter I got was from Carol Jennings, and she had drawn her family tree, described her family tree.
The breakthrough itself came in 1991. We got a letter in January telling us they had identified a gene that they believed caused this Alzheimer's disease.
When we found the mutations in amyloid in Carol's family, we knew that that is where the disease started. It's been a dominant feature of all work on Alzheimer's disease. Carol's family has what's called autosomal dominant Alzheimer's disease, and that means that if your father or mother has the disease, you have a 50% chance of inheriting the gene, and if you get the gene, you're going to get the disease at a very similar age probably to your parent.
She's a shell of the woman she once was, but she's still my wife, and so she's still part of who I am. We may not be able to change the cards that life deals with us, but we can change the game we play with the cards we've got so that future generations don't have to carry the same pain.
Yeah. Yes, Carol, this is the first family we found mutations, and I'll talk about that for a second, but I'll just say, really, this shows how much it's important that people with Alzheimer's disease and their relatives come forward and take part in research. Carol was really, and you got a little flavor of it there, you got a really important person, and she campaigned for research into Alzheimer's disease all her life. When we found the mutation, which I'll discuss very briefly in a minute, I have to say, when she found the mutation, I thought we'd have the disease sorted out within 5 years, and this was 1992. Carol was much more realistic, and she said that she didn't think the research would help her, but she hoped the research would help her children and her children's generation. And you briefly saw her son in one of those pictures, and her son is now going through clinical trials at Queen Square. So, and he's on the experimental drugs. So I think she was far more realistic than I was. She was really a remarkable woman, and I went to her funeral about 4 months ago, and at the funeral I found out that she'd been also supporting her local university, Nottingham University, by getting studentships for PhD students. So she was really, really a remarkable person, and I felt this film was made about 5 years ago, and when the film was made, I felt, of course, proud of the research we'd done, but I also felt it had been good research and we'd learnt a lot, but it hadn't led anywhere in terms of the patients. Now, I think we're beginning to see a change in that, and that's what I'm going to be talking about now. Do I do this? Yeah, I think. Now, this is the pathology of Alzheimer's disease. I'll explain it very quickly. Top left, you see a human brain cut down the middle, and you see here, I don't know if the mouse shows, but you can see the expanded ventricle. That's because you've lost brain tissue. Here, this is the hippocampus here. Yeah, it doesn't. maybe if I move it around a bit, it'll appear. Well, at the bottom of the cross-section of the brain on the top right, there is a hippocampus. That's where the brain, if you like, helps make new memories. And that's really shrunken in this cross-section. And that explains the earliest symptoms of the disease. The earliest symptoms of the disease are a problem in making new memories. It's often described as a memory problem, and that's true, but often, usually, in fact, people in the early stages of the disease, they can remember things that happened decades ago, but they can't remember things that they did this morning. The problem is making new memories. That is the first, those are the first problem. Those are the first symptoms. Bottom left, you can see a section through the cortex of someone with Alzheimer's disease, and those black flame-shaped things, they are tangles made up of the tau protein. In the bottom, in the middle, those are plaques made up of the amyloid beta protein. Those are about 1/10 of a millimeter across to give you some context of size. And bottom right is a blood vessel in Alzheimer's disease showing the amyloid, the same amyloid, around the blood vessel. That's called congophilic angiopathy. And actually, we've rather ignored that pathology to some extent until quite recently. But as I'll come back to at the very end of my lecture, that turns out to be very important. Now, what you're seeing here is a complicated pathology. And what you don't know is you don't know where that pathology started. So it's like coming across a car crash. You can see the car has crashed, but you don't know if the driver was going too fast, or the brakes were failed, or he swerved to avoid something. You can just see the final result. And I started my career by being a geneticist, a pathologist, a chemical pathologist, trying to look at the brain of someone with Alzheimer's disease. And then in 1983, the Gusella group in Harvard showed that you can locate genes for diseases by using then new genetic technologies, and localize the gene for Huntington's disease to chromosome 4. And for me, that was really a pivotal moment. I read the paper, and I thought, wow, this is so cool. You can find out how a disease starts, but through genetics. And so I changed my lab's goal. I'd just been fortunate to be hired in a genetics department. Actually, I've been hired to teach glycolysis and the TCA cycle. So this meeting has been really a trip back in time. Well, not back in time because there's been a lot of changes, but it's been interesting for me to see, to come back to my old area, if you like. Anyway, I started to study genetics with the help of my head of department, and started to try and follow, if you like, Gusella's recipe with Alzheimer's disease. So with seeing this pathology, there's obviously many ways the disease could start, and although the amyloid hypothesis is credited to me, or in the case of people who didn't like it, blamed on me, neither is really true, because in fact the original amyloid hypothesis is in these two papers from Glenner, top left, and from Masters, bottom right. Glenner in 1984 pulled off the blood vessels from an amyloid brain and sequenced the amyloid and got the sequence of the amyloid peptide. And then a year later, he got the sequence of the amyloid peptide from a case of Down syndrome. People with Down syndrome always get Alzheimer's disease when they get to be 50. He realized the sequence is the same. And so he wrote, you know, this shows to me, to Glenna that is, shows to me that people with Down syndrome probably get Alzheimer's disease because they make too much amyloid, and probably that's because the amyloid gene is on chromosome 21. So really, this paper from Glenna in 1984 really is the beginning of the amyloid hypothesis. Almost at exactly the same time, the paper from Colin Masters came to pretty much the same conclusion independently. Tragically, George Glenna died in the late 1990s. Colin Masters is still working and doing great work in Melbourne on the amyloid metabolism. So his work continues. George Glenna died, as I've just said, in the late 1990s. This was Carol Jennings' family. You can see here a grandfather with Alzheimer's disease, and then that grandfather had two sons, both of whom who got Alzheimer's disease, all of which at the age of about 55. Those two sons married two sisters. That's not important for this story, but it's true, so I put it in. And they've got 10 children. The SIB ship on the left have got 10 children. The SIB ship on the right have got 3 children. And what you're looking at is the top half of chromosome 21. How is the top half of chromosome 21 inherited? And you can see all the people in black have got the disease, and they've inherited all of the chromosome, as has their double cousin on the right. But 2 individuals, number 9 and number 11 on this diagram, have inherited, are not sick and did not get the disease, and they've inherited chromosome segments. And so the simplest way of interpreting this is that the amyloid gene is in the middle, and there it is, the APP gene. And that led us to sequence the amyloid gene and find a mutation. Now, this diagram, if I gave it to a good postdoc these days, this sort of family could be sorted out in probably 3 months because of the sequencing of the human genome. This was before, I don't know if anybody in the audience besides me remembers a time before PCR, but this was before PCR. And so even PCR was exciting. We didn't know the order of genes in the human genome. All of these things have come subsequently. So what you're looking at here is 30 person years of work. Nowadays it would be, yes, maybe 3 person months. And this is because of the changes in technology. The top paper is the paper, 1991. If you want, you know, when you tell a story in science, you always tell it as if it's been logical and everything's gone smoothly. Anybody, I should imagine everybody in this room, but certainly many of us will realize that it's rarely as smooth as you tell the story. And the dirt behind the story, if anybody is interested in the dirt behind the mistakes we made, that's in the bottom paper here. I would say that the main mistake we made, which I think was understandable, is that we assumed that all cases of early onset disease had mutations in the same gene. We thought that it was one gene for the disease. I think that was reasonable. That's true of Huntington's disease, which was our model. But for Alzheimer's disease, it's in fact only about 10 to 20% of early onset cases are caused by mutations in the amyloid gene. We did this, so this paper came out in 1991. As I've said, I felt that we geneticists were the referees behind which could adjudicate between all the different theories for the disease. So I wrote the paper top right and the bottom middle there, and Dennis Selko at the same time wrote a very similar article saying the amyloid hypothesis. And what I'll say about the amyloid hypothesis, before I criticize it, what I'll say about it is that it was simple enough for people to understand. And I think that's very important. And it was simple enough for the accountants who ran drug companies to understand, not just the scientists. And I think that was also important, so that it reached past the scientists. Anyway, so we found amyloid mutations in about 10% of the families that Martin Ross and my colleague had collected. And then there was obviously a race on to find the other genes involved in Alzheimer's disease. And that race was won by Peter Hislop, who was then in Toronto. He's now in New York. And he cloned this gene, presenilin 1, and also he cloned presenilin 2 at the same time. And the presenilin protein is drawn in blue there on the top left. And actually, and in red on that diagram, all the amino acid residues in red are places where we and others have found mutations. Now, the important thing about presenilins, which we realized afterwards because of work from Japan and work by Bart de Strooper and also from Dennis Selko, is that presenilin is the major component of the enzyme which cleaves the amyloid protein. So in yellow, we've drawn the amyloid protein in fact, what is the active site of presenilin, and presenilin cuts the amyloid protein. And the mutations in it usually alter the position of that cut and make the amyloid that's produced, if you have a mutation, marginally longer and therefore marginally less soluble. And that's dealt with in the paper at the bottom there. And so that's completely consistent with the amyloid hypothesis. The mutations in either amyloid or in the protease, which cleaves the amyloid precursor protein, are both causes of Alzheimer's disease. I'll take just an extra second or two to say what I've said before, in that these mutations are rare. And within the UK, there's only about 100 families with mutations in either the amyloid protein or the presenilin protein. So they're rare. And when we wrote the amyloid hypothesis, and there was a lot of criticism of it, but the major and really understandable criticism was that it was perhaps not correct to extrapolate from such a few cases of Alzheimer's disease, like I've said in the UK, just about 100 families, to the fact that within the UK, there's about 1 million people with Alzheimer's disease. A lot of people felt it was too much of an extrapolation to make that jump. And that, I think, was a fair criticism, which I'll deal with in a second. Now I'm just going to take a very brief diversion. We had been also collecting families with other dementias. And one set of families we collected were families with only tangles. And in fact, I had collected from an old colleague of mine in Brisbane, Australia, a large sheep farming family with this disease, where they got only tangles through the brain. And when we analysed these, we eventually found tau mutations. So tau mutations led to this other dementia, a tangle-only disease. We made the finding that our paper is on the left, but at the same time, Jerry Schellenberg, bottom right, and Maria Spillantini in the middle there also made the same finding. So we had amyloid and presenilin mutations cause full Alzheimer's disease. Tau mutations cause tangle-only disease. And what this enabled us to do, was to make mice lines. We'd made mice lines which had amyloid pathology already. Now, having found tau mutations, we made tau mice which had tangles. And so we crossed the two sets of mice And the best place to look is in this middle bit labeled entorhinal. What we found when we crossed the two sets of mice is that the mice with only tau mutations had very few tangles in the cortex, some but very few. But when we crossed them with the amyloid mice, it increased the amount of tangles to a large extent. So what this is telling you is that amyloid is upstream of tau in the pathologic cascade. Amyloid makes worse the tangle pathology, but tangles don't alter the amyloid pathology. So it's giving a direction to the pathology. And that allowed us to draw this diagram, which is, if you like, the diagram of the amyloid hypothesis, which is saying amyloid driven here by APP. Oh, sorry, I keep thinking you can see my amyloid on the left here, or presenilin mutations give rise to amyloid, and that drives on the right tau dysfunction and tangles. So this is a very simple well, obviously it's very simple-minded. Well, anyway, it's there. So it's I think this diagram has some truth to it, but it's also really worth looking at the problems with it, which are manifold. The first problem is, look at all those question marks. We drew this diagram in 1998, and actually none of those where all of those question marks are, there's still question marks. And the other problem is that we've got that line down the middle there, which I imply is a neuronal membrane. So I'm implying that amyloid on the outside of the cell is causing tangles on the inside of the neuron. So the only cell on here is the neuron. That's the only cell drawn in this diagram. Now, you know, I've said that I'd been hired to teach glycolysis in the TCA cycle. And actually, what I've been thinking was that we would sort out Alzheimer pathogenesis and draw a diagram rather similar to glycolysis, that this leads to this, leads to this. But as you guys have discussed a lot here this time, and I knew already to some extent because I was teaching it, if you alter glucose levels, you change the level of glycolytic and TCA cycle intermediates in milliseconds. It's really a biochemical pathway. It operates extremely quickly. And what we now know is that this process under this diagram takes 20 or 30 years. And I think a process which takes 20 or 30 years does really not fit into the same model as glycolysis. So that's a problem with this diagram. And I think it took us all quite a long time to realize that that was the case. Now, I've said already that the problem with the amyloid hypothesis, as you like, is that it applies directly only to a few cases with amyloid or presenilin mutations. What's going on in late onset Alzheimer's disease, the sort of Alzheimer's disease that every geriatrician, neurologist and geriatric psychiatrist sees every day? And we couldn't answer that question until the advent of the genome-wide technologies we now have, starting with genome-wide association studies and moving on now to whole genome sequencing. And the work that we did in this, all the group, well, I'll also say a little bit about the philosophy of genetics. What used to happen in genetics was that every group used to get their own families and compete. It was competition red in tooth and claw. We all got our own families. We were very secretive. We sequenced and analyzed our own families as fast as we could. It was no cooperation. You know, we were always trying to work out what the others were sequencing and so on. No cooperation. With genome-wide association studies, suddenly we had to all play nice in the sandpit, because what you need in genome-wide association studies is huge numbers, far more than any individual group could collect, and everybody needed to pool their data. So we all had to go from ruthless carnivores to vegans, basically, overnight, and that was what happened. We managed to do it. It wasn't without a few squabbles, but we managed to do it. And in the UK, our work was led by Julie Williams. In the States, it was largely led by Jerry Schellenberg, and in France by Jean-Charles Lambert. And we got together very large consortia. And what we did in those consortia is we ran about 500,000 genetic markers through, well, now several thousand Alzheimer cases and controls. Now, what you do when you do that is doing thousands of chi-squared tests, essentially. So you have to set your Bonferroni correction very high. So to explain this diagram, you've got the chromosomes and the markers along here. Sorry, I keep pretending you can see what I'm pointing at. Here, and you can see a faint red line, maybe about a quarter of the way up, and that is the Bonferroni correction. And you're allowed to declare any gene that comes above that line. And what you can see here is a lot of genes which come across the line. On the right-hand side, you can see ApoE. That's easily the most important of them, and I'll come back to that in a second. We had a very good lecture about ApoE today, which I was fascinated by, and then all the others come up. Now, what we had expected was that we would find lots of genes involved in amyloid production. That's what we had expected. You'll notice that this slide is headed not obviously amyloid related. You'll notice the subtlety of the italics there. That's because, in fact, they turned out to be amyloid related, but not in the way we saw. So these were the genes that we found, and Leslie Jones, sadly died last year, was the first person to realize what these were telling us, and she said, this is the title of her paper, Genetic Evidence Implicates the Immune System. By the immune system in terms of the brain, she largely, not completely, but largely means microglia and cholesterol metabolism. I would not write cholesterol metabolism now. I would write lipid metabolism, more general than cholesterol metabolism. So she pointed out that nearly all of these genes were either microglial or involved in lipid metabolism, or in fact both, microglial genes involved in lipid metabolism. And that, I have to say that when this was published, this was well, I can only speak for myself, this was a real surprise to me. I think it was a surprise to a lot of other people too. I'll take another little diversion now. I had been very aware of the fact we'd worked out autosomal dominant dementias. Were there any autosomal recessive dementias? And to try and sort this out, I had gone to visit collaborators in Turkey, where there is quite a lot of consanguineous marriages. Met up with Murat Emre and Ebba Lohman in Istanbul, and started to look through their collection of families with recessive disease, recessive dementias. And they picked out these 3 families, and they all had mutations. When Rita Guerrero in my lab sequenced them, they found families with mutations in a single gene, TREM2. I'll just read the mutations for a second, because I'll come back to that. The mutations were Q33X, that's obviously a loss of function mutation, T66M, and Y38C. Oh, thank you. Oh, great. Thanks for that. Here we go. So she had found these mutations in these 3 families, you'll notice this weird title of the paper. Exome sequencing reveals mutations in TREM2 presenting as a frontal temporal dementia syndrome without bone involvement. The reason for the weird title, which again I'll come back to, is that Leena Peltonen, then in Finland, now also sadly dead in the last 10 years in London, in Cambridge, had identified TREM2 mutations in Finnish and other families without bone involvement. And so we had not suspected this was the same syndrome because the phenotype was different. We did not have Paget's disease of bone in our family, in our 3 families. Here is the data that we had got. What do you do these days when you find a mutation? What you do is actually, you look in 2 places. You look in your own database. Have you ever found that mutation before? We're fortunate at Queen Square because a very forward-thinking neurologist called Anita Harding started to collect DNA samples from all the patients who came through Queen Square in 1991, I think. So we've now got a database of 40,000 neurological disease samples which have been sequenced. And you look also, though, in the general databases which are now online, for example, from UK Biobank and so forth. So Rita looked there. We had about 1,000 cases and 1,000 controls at the time, and she found that, in fact, these mutations had turned up in our Alzheimer cases, but not in our Alzheimer, not in the controls. We had 6 cases with the same TREM2 mutations, but only heterozygous, not homozygous, in the Alzheimer cases and not in controls. And then this mutation, R47H, which was found in 22 cases and only 5 controls. So this tells you that TREM2 is a risk gene for Alzheimer's disease. Homozygous mutations cause this rare syndrome. It's actually a white matter disease, leukodystrophy, and heterozygous mutations increase your risk of Alzheimer's disease by about 4-fold. Our paper is here, top left. At the same time, this paper came from the Icelandic group looking in the whole Icelandic population and identified R47H. So TREM2 is clearly a risk factor. When we wrote this up, we got a note. I got a very nice, polite note from Tom Bird in Seattle. He'd worked with Leena Peltonen and had given her one of the families in which TREM2 mutations had been found, and he had reported previously the pathology in that case and pointed out that they had plaques and tangles. So as his note noted, that in fact the data tying TREM2 to Alzheimer's disease have been there since 1983. So his note very cheerfully said, if you'd done this 40 years ago, you'd have beat me to publication. Now, at the same time that we were doing these genetic studies and finding TREM2 as a risk locus for Alzheimer's disease, we were also doing gene expression experiments. What we were doing was looking at mice with amyloid pathology and seeing how that altered gene expression in the mice, in the mice as they developed Alzheimer pathology. And really, remarkably to us at the time, TREM2 was the gene whose expression went up in the most when you started to develop plaque pathology. So we had 2 completely separate findings. TREM2 was an Alzheimer risk gene. TREM2 responded to amyloid deposition. So Mar Matarin and Dervis Saleh in our lab started to look at what other genes had their expression altered by amyloid deposition, and their their work is on here, and this here is TREM2. That is the gene whose expression is changed the most by amyloid, but these are the other genes whose expression is changed by amyloid, and those circled here are also Alzheimer risk genes. So what this is showing is that genetic variability to how well you respond to amyloid deposition is part of your risk of developing Alzheimer's disease. And that led Dervis Saleh in my lab to look more carefully and ask these questions. Is this a general principle? Is there an overrepresentation of amyloid to amyloid response in brains as amyloid is deposited? And the answer to that was yes. And is that still true when we take out the genes we already know? And the answer still is yes. And so then what other genes are part of this? And that is in the next slide here. All these genes underlined in blue are genes which had not been described before, which were also Alzheimer risk genes. So simply by looking for which other genes were responding to amyloid, we were able to find other risk genes. You remember I said about Bonferroni correction when I showed the GWAS. You know, Bonferroni is really a pain in the ass to any scientist. We really don't like him, but what this allows us to do is to cheat the Bonferroni correction a little bit. What we're doing here is instead of looking at all the genes in the genome, we're only looking at those genes whose expression is altered by amyloid deposition. So for those of you as statisticians, what we're doing here is an unsophisticated cheating on the Bonferroni correction. Anyway, that enabled us to choose the genes underlined in blue. I'll just mention one of them, though I'm not going to talk more generally, and that is the gene OAS1. My eyesight isn't quite good enough to point it out. There it is. OAS1, that's an amyloid response gene. That's part of the interferon pathway, and it's worth pointing out because exactly the same gene is important in the response to COVID. It isn't part of the COVID infection susceptibility. What it's important in is if you get COVID, how bad is it for you? OAS1 is part of that. And in fact, other genes, other interferon genes are a lot of that problem. So those of us who get COVID and respond badly have usually got a weaker interferon system. That's aside. But it's interesting that it shows, if you like, that there's a commonality between bad COVID and Alzheimer risk. Now we've gone on and followed this up more carefully. So we know all these genes are risk genes, and we know that they're amyloid responsive genes. So what we next have done, and this just came out about a year ago, what we've next have done is done experiments to understand using tissue sections when is gene expression changed. And the answer really is that most gene expression only changes when the microglia touch the amyloid plaque. Those of you who work, in fact, most of us know that what microglia do is they survey their environment all the time, and their gene expression only changes, and TREM2 is the leader for this. Their gene expression changes when a microglia touches a plaque, and then it changes its gene expression. So this is, if you like, the first stage of Alzheimer pathogenesis as plaques form. Now, one of the things we've been very interested in is understanding, having worked out the genetics of risk, to try and dissect that risk. And the public databases have really allowed us to do that. What I showed you before was the GWAS for clinically diagnosed Alzheimer's disease. And we have done a smaller GWAS, which is looking at pathologically diagnosed GWAS. Now there's public databases, for example, ADNI in the States and ABLE in Australia, where people have looked at amyloid positivity on PET and then done a GWAS on PET amyloid positivity. And what this has shown, which is a surprise, is that the genetic risk for Alzheimer's disease is slightly different in an important way from the genetic risk of amyloid deposition. Amyloid deposition is almost entirely ApoE dependent, but dementia in that context depends on the microglial response. So in other words, it's dissecting the pathogenesis into Alzheimer's disease. Amyloid deposition, nearly entirely ApoE dependent. Dementia in that context dependent actually largely on the microglial response to that deposition. So it separated the process into two stages. And there's this is a very nice paper that came out which comes to the same conclusion in mice. This group repeated the experiment I showed you where we crossed amyloid mice with tangle mice and showed that amyloid made more tangles. If you did this when you'd paralyze the microglia by knocking out the TREM2 gene, what you got was far more tangles. And so what this is saying is that the tangles, the microglia are protecting the brain from the consequences of amyloid deposition. And that, I think, is a very important idea. And it led me to come up with this suggestion based on these papers. So these two papers here. ABCA7 is an Alzheimer risk gene. That's a phospholipid transporter. And Colonna's group had shown that TREM2 is a phospholipid receptor. So this is pointing really at the lipid metabolism. But you know, when I saw that these were phospholipids, and ABCA7, which is also an Alzheimer risk gene, is a cholesterol. This is pointing at lipids, and to me, lipids means membranes. And I'll just discuss that and explain what I think it means. This is how we normally draw amyloid metabolism. You can find this diagram or a version of it in multiple places. I think it's also on Wikipedia. Here you have the amyloid precursor protein. This is the alpha pathway, which doesn't lead to amyloid deposition. This is the beta pathway, beta secretase cleaving here and presenilin gamma secretase cutting here. And you draw, and it draws amyloid going off into the extracellular space here. But anyone who works on A beta, especially on amyloid 42, knows how insoluble it is. It's a transmembrane domain. These are very, very lipophilic. So I think that this is the misleading part. I think drawing it going off into the extracellular space is the mistake in this diagram. And what I would suggest instead is that the beginning of amyloid deposition starts in the membrane. And this process, it goes on for a long time before you start to reach dementia. And what happens is that you get membrane damage that activates the microglia, and the microglia come in and save the situation. And actually, I was really interested by the lecture today showing that ApoE2 seemed to be a better lipid mover in terms of the brain than ApoE3, and better again than ApoE4. And I really I really want to follow that up and try and understand it, because I think that could be a very important finding. But what you're looking at, the reason the process, in my view, takes 20 years is that for that period of time, the microglia are saving the situation, and eventually they become overwhelmed. And I think this is a general principle for these late onset diseases. You have damage repair systems, here in this case it's ApoE and microglia coming up and removing damaged membranes, but as a more general principle, I think it's true too, that what you have with late onset disease in general is a failure of damage response. So this is where we're up to in terms of research. Early onset disease caused by too much amyloid, late onset disease predisposed to by poor clearance of amyloid. Now, having given you the justification for amyloid, why has it taken so long? And I think it's incumbent on us to say why things have taken so long. The first reason, I'm afraid, and it's dealt with in this paper that actually Eric Caron wrote at the bottom here, is a lot of the early trials which claimed to be anti-amyloid trials actually were not anti-amyloid trials. They were drugs, for example, they were drugs which didn't hit amyloid at all. They were drugs which were given at a dose less than 1% of the concentration needed to alter APP metabolism. So there was a lot of, well, bullshit in those early trials. So a lot of them, which, you know, what would happen is a company would say they were going to do an anti-amyloid trial, and then the trial would fail, and everyone would say, look, it's an anti-amyloid trial and it failed. But really it was a bit more complicated than that. And that was why a lot of them failed. That hasn't been true more recently. For example, this trial was a very well done trial of solanezumab by Lilly, and it also failed. Why did it fail? Well, what all of us had not realized was that many of the antibodies, they prevented amyloid deposition, but what they didn't do is suck amyloid out of the brain. So they prevented further buildup, but they didn't suck the amyloid that was already there out of the brain. And I don't think, well, again, speaking for myself, I had not realized how important that was. I think it was, you know, they were good trials, sensible trials, well carried out, and they prevented further amyloid buildup, but they did not suck amyloid out of the brain. Now, for me, this paper, Caron and Destrupa, which is in 2022, I think it's in Nature Drug Discovery, was really an important paper. And what this does is it gives their view on what is an important, how to attack amyloid. And it says using antibodies. And just looking at the diagram, this is theoretical. It says that you have to start giving the drug, it has to remove amyloid, and then you'll only start to get divergence of clinical symptoms from placebo when you remove the amyloid. And so it's saying the trial has to be long enough for you to clear amyloid, and then long enough after that time to see a clinical benefit. And that, for me, this was really an important paper. It's really, well, you know, what you get in science a lot is people who tell you what you did wrong after you'd done it. You get that a lot. And the nice thing about this paper is it came out and was written before the successful trials. So it predicted the future, not smart-assed the past, so to speak. So here we have now the real data from the trials that have been, which have been in the news and so on. So lecanemab first, you can see here, over, well, let's say at about 10 months, it's really reduced amyloid a long way. Donanemab, also equally successful. That's going through the approval circus at the moment. That also seems to work. Aducanumab is interesting because aducanumab had 2 trials, and the first trial showed significant effect and amyloid reduction and therefore clinical improvement, but the second trial did not work quite so well and did not show a benefit. And that led to a controversy, which you might have read about whether it should be approved or not. I have to say that if I'd had a vote, I'd have voted not for approval, because they have a rule 2 successful trials, and they only got 1. I think that was a mistake. Anyway, that's what happened. But it's an interesting biological thing that the trial which removed most amyloid actually did have better clinical outcome. And then thirdly, reported last year, the unsuccessful gantenerumab trial from Roche. Now, I'm showing gantenerumab now as an unsuccessful trial, and you can look at this and understand in the context of that review why it was unsuccessful, but actually, I think I'm going to come back to it in a second. So these are I'll just show you this for a second. These are the original reports of those trials. Lecanemab in New England Journal, impact factor about 50, completely successful, everyone agrees it. JAMA, I think impact factor about 15, second to come out, still very good results. Aducanumab, everybody argues about the result, published in a journal with an impact factor of about 3. So I think that shows the outcome. Now let's have a look at the lecanemab data. Here is the lecanemab data. I'm not going to show the donanemab data, not because of favoritism, but just because it's essentially exactly the same. And you can see here the rapid removal of amyloid and almost complete removal of amyloid over 12 months. This is using PET scans. Now, this is the clinical data which goes back to that. Now, what you can see from this clinical data is that there is a clear slowing. The trial has clearly been successful. But the other point to make is it has not stopped the progression. It's slowed it by about 25%. And all of the arguments, well, half of the arguments about whether it should be approved or not relate to the fact that it's only slowed progression. If you do a kind of back of envelope calculation, if somebody is going to go from diagnosis to needing nursing home care in 5 years, on this drug, the implication is they would go to nursing home care in about 7 years, not 5 years. So that's arguably, and if the argument is happening now, worth doing. And I think that obviously what we have to do is get better. Why are we not stopping disease? And there's a number of reasons why we might not be stopping. The first reason is that it might be that the tangle pathology has taken over, and the tangle pathology is continuing to build up, and that is driving the continued slope. That could be part of it. The other part of it is that perhaps we just did not remove the amyloid early enough, and we need to get the drug in earlier. So and the final thing I think is really important is we don't know what the pathology of these people who've had lecanemab is after they die. Have we removed all the amyloid? Are there some parts of the brain where there is still some amyloid? What is the underlying pathology? Is it, for example, that they have got other pathologies there, like vascular pathology or TDP43 pathology? Are we missing those? Is multiple comorbidity the issue? We don't know. We won't know the issue until we get neuropathological investigations. Very important. Can we get better? Oh, and the other point, and that's the first line here. The other point in the argument about approvals is ARIA. ARIA stands for amyloid-related imaging abnormality. The first thing, when you give an antibody, and it's an anti-amyloid antibody, the first thing it hits is that amyloid in the blood vessels. That's the first thing it hits. And so you get an inflammatory response on the blood vessels as the antibody comes through at high concentration and hits that amyloid. The amyloid in the plaques is behind the blood-brain barrier, but the amyloid in the blood vessels is there and in the line of fire. And so that's the problem. Now, usually that has either no patient-related effects. Sometimes it gives the patients headaches, but just occasionally it exposes hemorrhages and leads to hemorrhages. And some of those hemorrhages, especially in people who are on anti-clot-busting drugs, can be fatal. It's rare, but it is a serious side effect. And what that means is that when these drugs are given, they need to be monitored at the moment by MRI and continuously monitored. So these are expensive drugs to give. So the cost, I don't know what the exact cost is, and it's probably going to differ between countries. The costs in the US are about $25,000 for the drug, and they reckon it's about $25,000 to administer the drug and check its safety for a year. And so this is a serious amount of money. So that's a problem. Can we get better? Yes, I think we can get better. We can get better by making earlier diagnoses. If we make earlier diagnoses, especially if we can get diagnoses before we get the amyloid deposition in the blood vessels, we might be able to both reduce the side effects of the drug as well as prevent amyloid buildup from the very start. So can we get better by diagnostic improvements? Yes, I think we can. But this is really going to be a difficult issue in every country. Rolling biomarkers out to all clinics and helping all clinics make an early and accurate diagnosis is a real, well, in the UK and I'm sure worldwide, is a real issue, and that is something that we need to work on. And we can get better also, of course, by treating other elements of the pathology, and there's very nice work on anti-tau therapies going through phase 2 trials at the moment. So we can get better. This repeats what I've already said. So these are the things we need to do: treat earlier, and we need to treat earlier, basically. Treat earlier, and also, though, see if we can avoid the side effects of the drug. And with that in mind, I think this is really a hopeful sign. So what this is the gantenerumab that failed. Gantenerumab failed. It wasn't fast. It did remove amyloid, but not fast enough or far enough. And it also, they had ARIA. What they have now done is tie the active site of the antibody, of the gantenerumab antibody, to the transferrin shuttle. So now the transferrin shuttle takes the antibody active site across the blood-brain barrier and seems to avoid the ARIA side effect. I think this is the most hopeful. This is Roche, but Biogen have got a similar approach too, and so have Denali. So these shuttles, which take drugs across the blood-brain barrier, which frankly I would never have believed would work, really are working. And you can see, you remember that gantenerumab had not really reduced amyloid sufficiently in 12 months, But here you see, when it's tied to the shuttle, it removes the amyloid in these, it's very few patients, so we have to be a bit careful, in about 3 months. So good times are coming. So where do I think we are? Well, and I also want to say one more thing, as I think it's important, and I said it in the discussion a little while ago. What we're talking about here is mechanistic therapies for Alzheimer's disease. I think good times are coming. But the other thing which has been perhaps more important so far is public health stuff, reducing high blood pressure, reducing cholesterol, reducing smoking. It seems as if those public health measures have reduced the age-specific incidence of Alzheimer's disease by 20 to 30% over the last 30 years. So we haven't made a fuss about it. It wasn't, of course, us neuroscientists who really did it. It was the cardiologists. But let's take the credit for that too. You know, science is reducing the incidence of dementia. And now I think we have mechanistic therapies which are beginning to work. So where do I think we are in terms of mechanistic therapies? What the analogy I give is that in 1907, the Wright brothers, with a modified bicycle, flew for a mile in a figure of 8 around a North Carolina beach. Who would have believed that less than 15 years later, you would be operating commercial flights between London and Paris? I think that, you know, once something has been achieved once, it's much easier to achieve it a second time and do better, and a third time, and a fourth time. So I think that, you know, it's been a disappointment that the EMA have not approved outside these therapies, at least lecanemab. I'm disappointed. I can understand why, given what I've said about ARIA and the expense. But I think this is a temporary setback. Good times are coming, I think. I think we are getting better. But there's a song which I also like, which is a Neil Young song, which says, good times are coming, but they're sure coming slow. Well, I think they're coming a bit faster now. And so we're in a golden era. Thank you very much. I just want to make one more point, and that is that what drives the research, of course, is the patient need. And that patient need is driven through the political process. And so I'm very grateful that the President was part of this evening, and the President's husband, who used to work with the pharmaceutical industry, I just found out, is here. I think that it's very important for all of us to have the political support to drive our research. We sometimes take our politicians for granted. We sometimes say things about them we shouldn't, but we should be grateful for their support. Thanks very much.
We kindly thank our esteemed guest, Professor Sir John Hardy, for his insightful, hopeful, future-oriented lecture. We've thoroughly enjoyed it. Ladies and gentlemen, esteemed speakers, participants, viewers of today's public forum on Alzheimer's disease, let us round off the closing event of the 15th International Conference on Brain Energy Metabolism, ICBEM 2024, with a summary of its main insights. Recently, the FDA, Food and Drug Administration, but not the EMA, European Medicines Agency, approved the first new drugs in 25 years to treat Alzheimer's disease, specifically antibodies targeting beta amyloid, a hallmark of neurodegeneration and brain energy impairment. Although these drugs are not ideal, they have renewed the commitment of researchers to develop more effective therapies. At ICBEM 2024, scientists and clinicians have come together to share and discuss research on pathways and cell-specific processes, including early impairments in energy metabolism that contribute to neurodegeneration. We hope that this meeting will help reshape the field by addressing age-related declines in brain energy and pathological changes, ultimately stimulating the development of new therapies with small molecules targeting age-related brain alterations. And with this, we have brought today's public forum on Alzheimer's disease to the end. We are pleased it has contributed to the furthering of mutual cooperation in the highly relevant field of neuroscientific research at the time of its unprecedented progress. We wish you a relaxed evening. We invite you to the reception awaiting us in the neighboring lobby, where we shall pleasantly conclude our reunion. Thank you for your attention, and let us toast to new neuroscientific breakthroughs to come. Thank you.